So good morning, everyone. I am delighted to see so many of you here. We've come to think of this as one of the most interesting and important meetings that we put on during the year. It gives us an opportunity to speak with you, gives all of you an opportunity to see who we are and what we're doing. Now, this meeting, it's evolved over the years, and one of the very nice ways it's evolved is that instead of us just sitting inside the Beltway in D.C. thinking about what you all might want to hear, we've put together a planning group to help us with this meeting, and got a lot of suggestions for things we might not have thought of including, and some advice about what we shouldn't be doing that we have been doing. Nice advice. So I just want to take a minute to recognize the people who served on that planning group. Ron Bartek. Ron, if you want to stand up. Paul Gross. Gardner Lapham. Amy Rick. Cynthia Rothblum-Oviatt. Dean Sir, and Tina Takarshruski, and then a whole series of NINDS people. So if you'd be interested in the future in helping us with the planning, talk to Marion. And this being on the planning group won't be a life sentence for those of you who are on it, and we will rotate membership. And we also, if you have ideas, you're not on the planning group, but you have ideas about things that you'd like to see, that would be great to hear as well. So I'm going to start off talking about some NIH-wide things. One of the things that I don't think NIH does a very good job of is telling the citizens of this country what NIH does and the impact that NIH has. It turns out that only about 7 to 8% of if you do a poll, and a number of organizations have done polls, if you poll the U.S. citizenry, only about 7 or 8% of the people in this country actually know what NIH, or the National Institutes of Health, is. And even those people who do know often don't have a really good understanding of what the impact of the taxpayers' dollars that we've gotten has been over time. And there's a wonderful part of the NIH website, which is cut off, but you can Google it by NIH Impact, and you'll come up with a page, and it lists under various categories what some of the impact has been. And at the top of the list is impact on health of people in this country. And I picked out two particular pieces to highlight. The first is that death rates from heart attack and stroke have decreased by 60% in the past 50 years. And a big piece of that is, for heart attack and stroke, is recognition of the importance of controlling hypertension and lipid levels. So that's based on NIH research, NIH clinical trials, the Framingham study, but a significant impact, 60% reduction death rates from heart attack and stroke. And a big contribution to that is, in fact, stroke. And then the other fact that's up there, among others, is that in the past 25 years, the proportion of older people with chronic disabilities has fallen by almost a third, so that people are living longer and they're living healthier time. And there are other data examples. And then the second piece after health is economic impact. That money that we get from taxpayers doesn't just disappear into a vacuum. It's actually an investment in the economic health of this country. So turns out that if you count up all the people who are supported by NIH, and that's not just inside the Beltway, it's across the whole country, we support 432,000 jobs. It's a pretty remarkable number of jobs. And for every dollar of NIH funding that goes into a community, it actually generates more than $2 in the local economy. And the one that's the most amazing fact in my mind is that last year we generated $62 billion in economic activity in 2011. So that's a twofold return on the investment. So health, economy, and then, of course, science and scientific discovery. And one of the things that NIH likes to brag about is that 135 NIH-funded investigators have received Nobel Prizes. And that's kind of in the past, but just last week there was this wonderful set of findings that was released, top right hand above the fold in the New York Times. It was, I think, on the left in the Washington Post, that a project that the NHGRI, National Human Genome Research Institute, had supported called ENCODE, Encyclopedia of DNA Elements, found out what the 80% of our genome that had been called junk DNA or dark matter does. And that's where all the switches are that control gene expression. So people have been totally focused on the genes that made proteins and didn't really worry about what the rest of the genome did. And it turns out that that's where all the controls are. So impact of NIH. And as part of an effort to increase awareness of what NIH does, we just had this last weekend something called a Celebration of Science. And this was a collaboration between Michael Milken and the NIH. It was a three-day extravaganza, part of which was on the NIH campus, also sponsored by Faster Cures. And I don't know why the URLs come out as this incredibly pale blue. I couldn't fix it. But there was a whole session on the NIH campus which had Francis Collins wandering around the campus with being filmed and people talking in Natcher Auditorium. And there were about 700 people there. And one of the nice things was that they actually recorded the whole session. And those recordings for the NIH component are again up on the website. And you can Google this by doing NIH Celebration of Science, and they have the morning session and the afternoon session. And there's some wonderful, there's several patients who had AIDS where it would have been a death sentence who are now alive and well. One of the things that made me very happy was that almost all of the sessions included some brain sciences. So we've often felt that the brain doesn't get enough recognition or publicity or money, to be honest, but that this Celebration of Science featured the genomics, personal genomics piece, featured a set of twins whose whole genomes had been sequenced and who had actually been cured from a dystonia that had puzzled physicians. There's a wonderful session on imaging the living brain. So it's well worth your time to look at it. So that's the impact of NIH and a little bit of stuff you could watch that tells you what NIH does. The piece or the biggest change at NIH since our last meeting has been the creation of the National Center for Advancing Translational Sciences, or NCATS. It was finally became a reality. It's been discussed for almost a year and a half, became a reality in December 2012. And the mission of this center is to really catalyze the development of innovative methods and technologies that will enhance the development, testing, and implementation of diagnostics and therapeutics across a wide range of human diseases. Now, all the institutes at NIH actually have programs within their own institute to develop devices, develop better drugs, develop better treatments, and to test them. But this new institute is going to be looking at not specific diseases or treatments or therapies, but actually look at the whole process of drug development initially and eventually device development. So it turns out as I'm sure many of you have been reading in the papers, it's an incredibly inefficient process. So very few successes, very expensive. And the hope is that this center will actually be able to derive general principles that will make it work better. Why NCATs, challenges and opportunities. Genes are being discovered every day that cause diseases, and it's particularly true for the neurological disorders. Everybody here in this room knows that there are huge unmet needs for treatments for many conditions, especially rare and neglected diseases, and a lot of those affect the nervous system. And this is the point I've already made about looking at the drug development pipeline as a process and try to process engineer it in a better way. Now, I would say I bet the pharma companies think they're doing just fine and can manage this on their own, but each company actually has need to meet stockholders' expectations, and the NIH doesn't have to do that. So we can step back and say, okay, how can we make the process better? And Francis Collins wrote a wonderful editorial about the rationale for creating NCATs and explaining some of the motivations. Now, we didn't get any new money to do this. So what was done was to take existing programs from several places, the Office of the Director and also the National Center for Research Resources, and put them together to make a new entity. And by bringing them together, there'll be much more synergy. So it includes Clinical and Translational Science Awards. There's 60 of these across the country. The NIH Chemical Genomics Center, which is doing high-throughput screening, therapeutics for rare and neglected diseases, the TREND program, and some of you may have been interacting with the TREND program. Bridging interventional development gaps. This is a source of funding for people who need funds to get an IND and to actually get into the clinic, and FDA, NIH regulatory science. It's clear that the FDA and the NIH work together to have a better understanding of the science that underlies decisions that the FDA is making about what's the best way to run a clinical trial. What does a biomarker mean? What do you do if you want to test multiple therapeutics at once? I mean, the old idea was you had to do them one at a time. So that kind of regulatory science, and then something called the Cures Acceleration Network, which is money set aside to do really innovative experiments in facilitating or accelerating progress. Now, I want to give you two examples of the kinds of things and programs that NCATS is doing, just to give you a flavor for what all the jargon means. One of the programs that has been created is something called Tissues on a Chip. And this is a program, big program, $150 million over 5 years. And a piece of it's being done by DARPA, people who brought you the internet. And DARPA is going to be engineering chips that will allow the creation of tissues, organs, like a little heart on a chip, or part of a brain on a chip, or part of an eye on a chip, using whatever kinds of cells they have in their freezers. NIH's contribution is going to be to allow the development of the right kinds of cells. So instead of using a liver cell line, NIH is going to fund applications that will create real hepatocytes, liver cells, or instead of a neuroblastoma brain tumor line, how to get real neurons or how to get real astrocytes. And so DARPA and NIH investigators will be working very closely together. And the FDA, which doesn't have a lot of money, is actually going to be working with the whole program so that when these become a reality, the FDA will approve them for testing toxicity. You know, right now, toxicity is often tested in animals, which dogs, mice, rats, and long term. So imagine if you could test heart toxicity in a week using one of these hearts on a chip, or liver toxicity in a week by using one of these livers on a chip. So it's a pretty amazing opportunity. And not only could you do toxicity, but you could also test efficacy of potential drug candidates before you took them into people. And The pictures on the bottom are actually from a project of a Dr. Ingbar, who's putting together a heart-lung chip micro machine and has made remarkable progress doing this. And Mark Sutherland and Dan Tagley, who are actually were at the time NINDS program directors, had a major role in this. Dan has, unfortunately for us, been recruited to NCAT. So, and then a second example of what NCATS is doing is trying to rescue drugs. Now, pharma will design a drug for a specific application and may spend years and a lot of money, hundreds of millions of dollars, to get a drug to the point where it can be tested for the indication that they've designed it for. But it may turn out that it's not efficacious for that particular disease. And so they basically say, okay, that's too bad. Guess we guessed wrong, and put that on the shelf. It can have been, through the FDA, be found to be perfectly safe, just not useful for a particular disease. So the thought was, how could NIH get those drugs out of the freezers and closets and actually see if they work for other diseases, rare diseases that pharma might not have an interest in? So they've worked very hard, and 8 companies have contributed 58 compounds to this purpose, and it's a several-stage review process. So people sitting in laboratories all across the country could look at those 58 compounds with the minimal information that was available on the web and say, gee, from what I know about the progression of Friedreich's ataxia or a lysosomal storage disease or Parkinson's, this might work. So then they would wrote up a very brief application and sent it in, and those will be looked at to see how likely it is that they'd work for the new indication. And if it looks promising, then they'll be invited to write a long application, and there's $20 million set aside to pursue all the most promising of these. And for us, what's exciting is that 14 out of the 158 applications are specific for diseases that NINDS has to worry about. So that's NCATS. Watch that space. Be very interesting to see what NCATS accomplishes. And again, there's a beautiful website that they've put up. And if you Google NCATS, NIH, you'll come up with that website. Lots of interesting programs. Now at NINDS, probably the most exciting thing we've done in the last year and a half is to create a phase 2 clinical trials network called NeuroNext. NINDS has traditionally has focused on large phase 3 clinical trials, big gambles, $40, $50, $60 million, and they don't always work. And we thought that, actually Petra Kaufman, who was responsible for doing this, really thought that the Institute would be better off if we looked at things in phase 2 kind trials, earlier trials, kind of check them out to see, was there evidence that this would actually be a successful intervention? So the goal is to accelerate drug development, and we hope to be working with private entities, although we don't have anyone signed up for this. It looks at all neurological disorders, so it's not a Parkinson's network or a stroke network or a lysosomal storage disease. It will take all diseases, pediatric and adult, which we believe will be much more efficient. And probably one of the most innovative things is it's going to use one institutional review board. So any of you who've been involved in clinical trials know that if you have a multi-center clinical trial, each site wants their IRB to look at the protocol. And if you have 20, 30, 40, 50, 100 IRBs, each IRB feels obligated to find something wrong in the protocol, and then it has to go back a change has to go through all the others. So it can take 2 years to get a protocol through the multitude of IRBs. And we managed, Petra just said, preference for funding will be given to those of you who are willing to use a central IRB. And we got a lot of applications, and everyone agreed to the central IRB. And we think that that's going to make a huge difference in the amount of time. And since time is money and time is brain cells, that's going to be huge. And the other thing we did was, that Petra did, I shouldn't say we, was to have standard master trial agreements. So NINDS doesn't have to negotiate with each of the sites for how much they're going to get for running the trial. So between the IRB and the site agreements, we're hoping it'll be protocol to patient in 2 or 3 months. And we're just taking it for a test drive. We have made changes that we think will help engage industry, and it builds on the strengths of the CTSAs. These are the NCAT's Clinical and Translational Science Awards, and this is a map of where those sites are. Now, we can have a great clinical trials network that's efficient and fast and able to move things quickly, but it isn't going to work if we can't recruit patients. Now, some of our communities, I'll take Batten's disease as an example, phase 2 clinical trial in Batten's disease, and within 2 or 3 weeks of the announcement of the trial, we had all the patients signed up. But this is very unusual. Often, NIH trials can fail because of a failure to enroll. NCI, something like over half of their trials get closed out because they haven't recruited enough patients. Huge waste of money, of time, and of the patients who've participated. So NIH recognizes that this is a significant problem, and they've created a website, and Marion Emmer was a part of the team that put this website together to educate patients about clinical trials. What does it mean to be a participant? What's the value of participating? There are studies that have shown that if you participate in a clinical trial, even if you're in the placebo group, you do better than if you don't participate in a clinical trial. And it includes some patient investigator stories, including some stories from NINDS, a very moving story from a Parkinson's patient. If you can't read the blue URL, you can Google NIH clinical trials and you'll come up with it. And not enough just to have a website, we also are beginning now to include a patient or patient advocate in our clinical trial advisory committees. And the thought here is that we may not be aware, I won't say sensitive enough, aware enough of the issues for patients about participating in the trial, and that this would be good for us. And if there's enthusiasm for the trial, that person can help us communicate to the patient community what the trial is about. And I'm going to finish up with a brief discussion of the NIH budget. The fiscal year starts October 1st. That's just like around the corner. While in principle, Congress is supposed to have passed budgets for all the parts of the federal government by October 1st. It hasn't happened in a long time. The solution sort of to that is to have a continuing resolution. And we've had, one year we had, I think, 8 continuing resolutions before we finally got a budget. And it was brinksmanship every time. Sometimes it got signed at midnight the day before. When I first started here at NIH, there was actually no continuing resolution passed, and it shut down the government, and I was told I couldn't come to work, which I thought was pretty crazy. Anyway, so I heard on the radio coming over that there's going to be a continuing resolution passed sometime in the next few days that will last until April 1st. That's 6 months. That's half our fiscal year. And the impact of a continuing resolution is we will get the same amount of money we had last year, which is good. On the other hand, we're in principle not supposed to start any new programs. It's continuing what we did before. And the discussion on the radio, NPR, was that the Defense Department will have to keep making things that they thought they'd finished making and can't start making new things. Anyway, so NIH may be a little bit more flexible. So there's a continuing resolution coming. Now, March means the election will have occurred, that the president will be in place, whoever that is, and that the new Congress people will be in place, and there will be several months to sort out the consequences of the elections. Now, many of you who actually fund investigators have probably heard that money's really tight. And one of the things that NIH and NINDS used to be able to do was to suggest to foundations who funded research investigators whose grants were regarded as really excellent but didn't make the pay line because money's tight. And then it turned out that we weren't permitted to do that. And nicely, the National Health Council and NIH have created a database of projects looking for funding. Again, there's the URL, and that one's black, so you can probably read it. But you can Google National Health Council, and you will end up finding this. And the goal is, if you're interested in a rare disease, and there's someone who has written in an application which and you can have access, if the investigator allows you, to the reviews, you could pick up this grant instead of having to do your own solicitation and your own review. It's kind of like pre-reviewed grants that you can think about. So I think it's a useful resource. I haven't tried to use it because I'm not an investigator looking for money, but if any of you have used it, I would be really interested in hearing how well you think it works. So I think we have time for a couple of questions for me before we move on to the next section. Yeah.
You mentioned Neuranext for drugs, but does it also apply to devices?
Petra. So Petra Kaufman, who is in charge of the program, is in the back. Petra, you want to?
Neuranext can evaluate any promising new therapeutics in a phase 2, in an early phase clinical trial, whether the therapeutic is a drug or a device. Neuranext is positioned to do either of these. The other good thing about Neuranext is that it's not disease specific, so it can take advantage of opportunities as they arise in different disease areas. Neuranext also has access to a broad patient population, ranging from pediatric patients to adult patients. So really any promising therapeutic that could be evaluated in a scientifically sound early phase trial, ideally a biomarker-informed trial, would be appropriate for Neuranext.
Okay, so the next person who's going to speak is Dr. Robert Finkelstein. He has served as the director of the NINDS Division of Extramural Research from 2004. He's responsible for a very long list of things. Planning, policy, coordinating scientific programs funded by NINDS, overseeing scientific review, grants management, and administrative services of the extramural program. And what he's going to do is to talk to you all about how to interact with NIH. Now, some of you in the audience know a lot about interacting with NIH and probably can add or comment on some of the things he's going to raise. Others of you may not have known what's the best way to interact with NIH and NINDS, and hopefully this will be informative. Great. Thanks, Bob.
So I'm going to show about 15 minutes worth of slides on this theme, but if you remember one thing, it's that we are really here to work with you. That's really the message. There's really no other message in what I'm going to say besides that. In my part of the extramural program, and I'll talk about what that means in a minute, we have some really terrific staff. In particular, we have, in terms of people who you'll be interacting with, we have program directors and program analysts. I have had various jobs throughout my life, as like a record salesman or running an after-school center, but I have never worked with a better group of people, and I'm proud to work with the people with whom I work. So, and these people are really here to work in partnership with you. So one thing I noticed is that, speaking of partnerships and contacting us, I forgot to put my email address on this slide. So I deliberately created a very simple email address when I started here. It's just Bob F, as in Bob Finkelstein, at NIH.gov. So I'll say that again, Bob F at NIH.gov. I would really encourage you to contact me with any questions you have. I can hopefully help you in a number of ways. I can direct you to the right people to talk to about your specific disease area. I can answer any questions about the way this place works, which is incredibly complex and arcane to any outside viewer. And any problems that you have, if you can't get any need that you have addressed, I am happy to talk to you about. I'm busy, you're busy, but you are my highest priority. So I'd encourage you to contact me with any questions. Now, in terms of things that Marion wanted me to say, I'm going to say them now because otherwise I'll forget about them as I give my talk. In the inside cover of your booklet is a fold-out card that actually has a listing of all our extramural staff, and I think some of our other staff as well. It's sort of like an index card with lots of names on it. So that is your guide to how to contact us. Also, Marion's office issues a really terrific newsletter about once a month, and if you're not already on her distribution list, you can sign up for that. This newsletter has updates about things that we're doing, updates about new initiatives from Francis Collins and the Office of the Director. So there's all kinds of interesting stuff in there that I encourage you also to look at. So I guess I advanced the slides here. Okay. So I think that's all I was supposed to say. Okay, so let me just very briefly talk about the way the Institute is structured. That's about as simple as I can make it. Is this okay, this is it. So basically, about 90% of our budget for the last couple of years has been about $1.6 billion per year. And about 90% of that money goes into funding extramural research. So these are labs around the country, investigators who work on diseases, who do basic research, translational research, etc. And then roughly 10% of the money goes on funding the labs that are here in Bethesda. So this is the simple view. It looks sort of like a Pac-Man image, but of extramural eating intramural or something, but that's not what it's intended to be. Now, by the way, I'm going to, in a minute, talk about my part of extramural, but I don't want to forget to say that in addition to the Division of Extramural Research, which I'm the director of, Alan Willard sitting over here as the deputy director, we also have three other very important parts of intramural. We have the Office of Clinical Research, headed by Petra Kaufman, who you just heard from, the Office of Translational Research, headed by Rajesh, who I think is here, but I don't see him. There he is, Rajesh Ranganathan. And we have the Office of Special Programs on Diversity, headed by Al Gordon. So those are the four components of extramural. Okay, our institute has a very broad responsibility. This is our mission, to reduce the burden of neurological disease. And one of the things that makes this kind of a very daunting mission is that there are over 600 neurological diseases that we're responsible for. And that's something that's important to keep in mind, particularly when you're talking to your program director. Most of the program directors are responsible for a broad swath, both of basic neuroscience and also of diseases. Now, we have this broad responsibility, but as Story mentioned, unfortunately, money is not increasing the way in an ideal world it would be. So this top curve shows our federal appropriations in millions of dollars. And NIH had its budget doubled between 1998 and about 2003. So you can see that the dollars were increasing steeply. And then this is what happened after that. But if you actually look at inflation-adjusted dollars, after the budget ended, this is what we were really getting, okay? So if you actually look at these numbers, you'll see that gradually the budget doubling is being undone. And we're worried that that trend could continue. So this is my part of the Institute that I mentioned. I'm not going to go through all these components. These are down here are some of the administrative components that I oversee. I oversee a lot of the nuts and bolts operation of the Institute, the grants management branch, which actually gets awards out the door. Ruth Lynn helps run our council meetings. We have an administrative services branch. But I also oversee the 6 scientific program clusters, which are organized around scientific themes that I'll show you in a minute. But associated with each of those scientific themes are various diseases that are related to those themes. We also have a review branch headed by Ernie Lyons that reviews a subset of the grants that come in, program project grants, a lot of our translational grants, which will be of direct interest to many of you. We have a training office, which also includes workforce diversity, and we have an Office of International Activity. So this is sort of my shop, DER, DER. Okay, these are the 6 program clusters. Now, in each of these program clusters, there are usually about 5 program directors. There are program analysts who work with them. So these are scientists. There are people who have PhDs and beyond who are experts in particular areas of neuroscience and neurological disorders. So I'm not going to go through all of these, but for example, the neurodegeneration cluster not only oversees research on cell death, for example, but it also oversees various diseases that are caused by cell death. So, for example, Parkinson's disease, ALS, SMA, Alzheimer's disease. I'll take one other example, and I'll use this because the name is not that transparent as some of these others. So the Neural Environment Group not only oversees research on neurons, but on the cells that are in the environment of neurons, like glial cells or vascular cells. This particular cluster oversees a lot of our stroke research, also multiple sclerosis, various contagious diseases, neuroAIDS. So these are the clusters that our staff, or at least my staff, are organized into. Okay, so this is the theme of my talk. How can we work together most effectively? And the first step is for you to contact us with any questions you have, and not just questions you have, but any insights or ideas you have. Okay, we don't want to become isolated, and we know that you are the experts, particularly from the patient's perspective, on the diseases that we're responsible for. So you often know far better than we do what the needs of patients are, what some of the research priorities should be if they aren't already. So in terms of how we can work together, I identified 4 broad themes. The first is to work with us to identify research opportunities. I'm going to go through all these themes in a little more detail, to find common themes across diseases, to capitalize on what each of us are good at, NINDS and you guys, and also to exchange information. So let's look at this identify opportunities topic. So basically, as I just mentioned, you, the patient advocates, have the best understanding of what the needs are for your respective diseases and disorders. And we can learn more about that by working with you through a variety of forums, either one-on-one conversations, workshops, many other forums. Understanding what these needs are is part of the way that we plan. We obviously talk to the extramural scientists who do the research, but in terms of coming up with strategic priorities and initiatives, we have often worked very closely with people in this audience and others to identify themes that we want to apply money towards. And there are various ways that we can interact. Only 2 of them are listed up here. In terms of common themes across diseases, this is becoming increasingly important. So I mentioned that we're responsible for a huge number of diseases. It's very hard for us to have a strategic plan for every single disease that we're responsible for, but we can have plans for groups of diseases which are often united by underlying scientific mechanisms. So for example, the simplest one to understand is one I mentioned a minute ago, neurodegeneration. There are many diseases that involve brain cells or cells elsewhere in the nervous system dying, and a lot of people think that the mechanisms that cause cell death may be very similar across all these diseases, and that what we learn in Alzheimer's can potentially be applied to Parkinson's. This is still something that we're learning more and more about, but we're attempting to have a lot of scientifically based mechanistic themes that we apply to diseases. This is also true of behavioral disorders, where phenomena like executive function may underlie a whole range of neurobehavioral diseases. Now, in addition to these scientific themes, researchers studying different diseases often need very similar resources. So in the past, for example, we funded a network of microarray centers that were used by investigators for researching a number of different diseases. We're also funding, through Rajesh's Office of Translational Research, the infrastructure for therapy development research. And here I'm talking about preclinical therapy development, everything that goes up to and including the investor getting an IND or an IDE from the FDA. So there are often across diseases, another common theme is that there are common obstacles in terms of both the science and therapy development. So an obvious one is that there's a drug delivery problem which is based directly on the blood-brain barrier. So we try to identify broad themes that we can focus on in addition to being aware of the various disorders that are affected by them. Okay, so the third bullet that I had was capitalizing on our respective strengths. So in terms of what you bring to the table, foundations and patient advocacy groups, you can often be more nimble than we can. We try to be as fast and agile as possible, but this is NIH and there are various processes that we need to go through to fund grants. But you can act very quickly in a disease-focused fashion. You can provide seed money for projects. A lot of our biggest successes in translational research have come from program directors working with you to fund very early phase research, which, if it's successful, can seed larger grants, translational research grants from our institute. You can help attract new investigators into working on your disease, and obviously you can and do stay continually in touch with patients. Now, in terms of what we bring to the table, obviously we have a really terrific peer review system, and this is the central strength of NIH. This is what makes NIH, I think, the best biomedical organization in the world. We have a very strong peer review system, and despite the fact that people complain about that system, particularly when the budget is so challenging, we bring this quality control element to the table in terms of evaluating research applications. We also help to create the pipeline that often leads to treatments for various diseases through funding basic research. We spend more than half our money funding basic research, and translational and, of course, clinical research. And included in the clinical research are, as Story mentioned, phase 2, phase 3 clinical trials, many of which would never be performed by industry. So we try to do things that are not redundant with what other people are doing. If industry is working on a particular clinical trial, that's not something that we'd emphasize. And particularly for many rare diseases, we may be the only game in town in terms of funding research. The next thing is the importance of exchanging information. We can, as Story mentioned, there's a website where we can inform investigators about other funding sources. So it's really very troubling to me when I see a really terrific research application that I know we can't fund because we don't have enough money. So if our pay line at one point was the 9th percentile, that means that we were really funding just the top, this is an oversimplification, but we were funding roughly the top 9% of investigator-initiated or targeted applications. And that really is a shame. So there are really strong projects that if you're interested, you can fund. So there are websites and ways that we can make you aware of some of the things that we can fund. Another thing, back when I was a program director, that was very important is that in various ways we can make sure that we're not doing redundant things. So if there's something that we're really focusing on, you may not want to focus on that. You may want to try to capitalize on it, or vice versa. If there's some initiative you're doing, it doesn't really make sense for us to do. If you're setting up a patient registry, it doesn't make sense for us to set up a patient registry. So I think it's important to work together to avoid redundancy. There have been an increasing number of public-private partnerships. That's a whole area that I'm not going to talk about in detail, but we've had initiatives that have been done in collaboration with some of the people in this room. And particularly in times when the funding climate is very challenging, this is becoming more important. It doesn't always involve your organizations giving money. In many cases, it involves you giving us expertise. Story mentioned that you can help us with recruitment for clinical trials, which is often the rate-limiting step of the success of these trials. And there are many other ways that we can exchange information. So how do you learn more? I'm going to keep my talk relatively short. Really, there's only one thing I wanted to say, which is that you should feel free to work with us. How do you learn more about our institute? Well, first of all, you should check our website, which we're continually trying to improve. We're in the process of hiring a new webmaster, and we want to update it and make it more user-friendly. So you can learn about various programs that we're initiating, various resources that we have, and all of it in one form or another is on our website. If you have problems using our website, let me know about it, and we'll try to make it easier for you to use. One thing you can do, I'm what's called the Executive Secretary of the NINDS Council. I think you'll hear more about this in the next session, but after peer review, there's a second level of review. Some of you know about it because you've actually been on our council or are about to be in our council. So the council looks at applications from a higher level. Peer review looks at the specifics of the science and gives the application a score, but council then looks particularly at large-scale applications or applications with particular issues associated with them. And they'll often say to us things like, well, maybe you shouldn't fund this grant because you're already funding a lot of grants in that area, or vice versa. So council is the second level of review, and you're welcome to sit in on the open session of our council. It's actually coming up next Thursday. And you can find the specific information about when it meets and what the agenda is on the Federal Register notice, which comes out like every day, I think. So one thing you might want to consider doing is look at the agenda, particularly if there's something you're specifically interested in, show up at the meeting. You can talk to me if you have questions about what this involves. But I think you can learn a lot about how the Institute functions by watching its council work. And then after the open session, we go into closed session where we look confidentially at specific grant applications, which are obviously we can't disclose. You can also, as you'll hear about in a minute from Jim Onken, you can also learn about what is currently being funded in your disease area or in a particular area of science. There used to be a website called CRISP that a lot of you know about. It's now NIH Reporter, and you'll hear Jim talk about how to use Reporter and what it can do, which is actually quite remarkable. You can do things like, what is your geographic area of the country? What are you getting in terms of NINDS funds, et cetera. Finally, I already mentioned the nonprofit newsletter. So going back to what I said before, the gate for a lot of you will be to identify the program director whom you can work with. If that's not immediately obvious to you, just send me an email and I can help you find that out. Our program directors and our program analysts are incredibly talented, dedicated people. They're almost all extremely overworked, but their primary responsibility is to work with you guys. That's the most important thing they do, as well as working, of course, with the scientists who do the research. So you can discuss with them what your organization is doing, what you see as important research opportunities. You can ask their advice about everything from setting up a scientific advisory board to whatever. And as I mentioned, however, you should realize that your program director is not just responsible for your one disease. That person may be responsible. Dan Togley, who's who just left, is a good example. He was probably responsible for 75 different diseases. Okay, I think, so just in conclusion, this is not the most optimistic time in terms of the budget. But I really believe in this cliche that these challenging times present an opportunity. From our perspective, we've thought very hard about programs that we should shut down, either because they're finished or they're not working. So it is really an opportunity for us to become leaner, hopefully not meaner, but leaner. And basically, even in challenging times, there's a lot we can do together. In fact, some of the most important initiatives that we've started have been started in very challenging budget times. So that's really all I wanted to say, and feel free to contact me if you have any more questions.
Questions for Bob?
Okay, so I'll paraphrase that. Okay, so what you said was that, what I said, most of what we do is funding research. But what about other things? What about getting educating clinicians about particular diseases or advances in basic research? What other roles do we play besides simply funding research? So first of all, before I answer that, I want to say that although a lot of the stuff that we fund simply based on its score and peer review. So for example, if we set the pay line, which it's set at now at the 15th percentile, almost any application that gets a score better than the 15th percentile will be funded. But in addition to that, there's other stuff that we fund, okay? So we have various kinds of initiatives, some of which are on groups of diseases or areas of science. So we very actively plan and fund things that are related to particular diseases or particular areas. But in terms of other roles, our primary function is to fund research. There's no doubt about that. But we believe that research has a much higher chance of being successful if it's, number one, often interdisciplinary where that's necessary. And often if there's a segue between different communities and different parts of the research pipeline. So for example, when someone comes in with a therapy development application using animals, one of the criteria that we use is what will happen if this is successful in terms of it being handed off to clinicians or clinical trials. So we try to make connections between different communities. But we also have a whole lot of workshops and meetings where we get different parts of our constituency together. So we deliberately, I'm just thinking of one that I was involved in when I was a program director. So at the Child Neurology Society, we did a series of forums on different diseases. So for example, we ran a forum on a disease called Rett syndrome. And one of the main functions of that forum was to get a lot of the basic researchers to present what they were doing and the clinical researchers, but the audience was all neurologists, child neurologists. So part of the function of this was to educate them about cutting-edge research examples. And we often do it the other way around, bench to bedside and back. So we have lots of forums where we try to promote interaction. And the reason we do that is not simply as an FYI to these groups, but we think the research will be optimized and will be much more effective if the clinicians are aware of what the basic scientists are doing and vice versa. So I don't know if that's the answer to your question, but we have a whole bunch of forums, informal in terms of phone conversations, council, many different workshops that try to educate and bring together different constituencies.
So there's also a section of our website which is an alphabetical list of diseases. I don't know if transverse myelitis is on that. It is. Marion's saying yes. If you look at that, we're constantly updating those and would be grateful to have your input to the extent to which we reflect accurately the clinical manifestations and disease. And I would just like to emphasize, we probably run 40 workshops a year, and it's a little stochastic as to where we do a workshop, but if there's a real need in a particular area, or if there are related conditions that can be bundled together, we would certainly consider that. We don't, we're not the ANA. We're not the AAN. We don't run those sessions about getting your CME credits on particular diseases. But yeah, you want to follow up?
I just wanted to ask, so you gave the website for the source of research funding. What about the source? Where can we go to find out about these workshops ahead of time, and where are they listed?
We don't actually list those in any coherent fashion, and that's one of the website is the major portal to the outside, and we're in the process of having a lot of soul searching about the extent to which we, but there is a program director who is engaged in transverse myelitis. I can't tell you off the top of my head who it is. They may or may not be here, but that's a really good place to start. Walter, do you want to add anything? I see you sitting there looking like you might want to add something. Nope. Okay. So one of the things that Dr. Zerhouni did, who was the NIH director before Francis Collins, was to really make what we fund as transparent as possible to the broad community. And he created, or he put the money out for people to create something called the Reporter. It's a searchable database of the entire portfolio of NIH grants, contracts, and intramural programs. And I can tell you there was extraordinary resistance to the creation of this tool by NIH staff. It's computer-based, so it's not like program directors are going through and saying this is pain and this is Parkinson's. It's all done by computer algorithms, and you'll find some grants under Parkinson's, which you might say, oh my God, that's not Parkinson's. So it's not perfect because computers, the algorithms are made by people and the computers execute them, but it's an amazing tool. And if you've never used it, you got to go in there and do it. And so we've asked Jim Onken, who is the Special Assistant to the NIH Deputy Director for Extramural Research and the Chief Analyst in the Office of Extramural Research. This is his baby. He can make it sing in ways that I certainly can't, and he's going to give you a short tutorial on how to really get it to work for you. Jim, thanks so much for coming, Jim.
Good morning, and thank you. I'm assuming that some of you probably have used Reporter, and some of you haven't. So those of you who've used it, this is probably going to be too long a presentation. And those who haven't used it, this is going to go too quickly, and no one's going to be happy. But I am going to go fairly quickly because really the best way to learn about Reporter is to go out and use it. And all I want to do is basically give you an quick overview of the kinds of things that are out there, and then you can go out and explore it more yourself. Just by way of background, as Dr. Landis mentioned, we started this project back in 2008, in part in response to the NIH Reform Act of 2006, which is NIH's authorizing legislation, which stipulated that NIH would make available on the web all of the research that we support and link it to other information such as publications and patents that have resulted from that research. And so we took what essentially had been the CRISP database a long time ago, and now are taking those data, those information on the research projects we support, and linking it with various different things to tell you more about that research and try to address, as Dr. Landis mentioned, better communicate what the impact of NIH funding has been and what the public is getting for their tax dollars. Since 2008, we've grown quite a bit. These are all the new features that have been added each year. And obviously, I can't go through all of these things, so just a few examples of what's available on Reporter. This is the report homepage at report.nih.gov. There's a quick links section. You can go to quick links. These are the most popular tools that are on the website, one of which is Reporter. This is the database of all the research projects we support. This is what the Reporter query screen looks like. At one time, it had a fairly logical layout. Now so many people have asked for new things to be added. I'm not sure that's still the case, but we're going to be redesigning this over the coming year. But there is still some sort of sense to it. Up in the upper left-hand corner is things to enter about the project that you want to search for. So you can enter text words that you want to search for in the text of the title or the abstract or the keywords that are associated with any project. Or because we have projects linked to the publications that result from the research, you can also search for text in the abstract of any publications. Or you can search for both text in the project and the publications. In the lower left-hand corner is information on the investigator and their organization. the grantee organization they work for, so you can look by investigator name or state or city or name of the institution. And then on the right-hand side is a bunch of what I call administrative kinds of data. What fiscal year was the project funded? What funding mechanism? Was it a center? Was it an R01 individual investigator-initiated project? Which funding opportunity announcement was the research funded under, and so forth. Dr. Landis mentioned our computerized categorization of projects. These are referred to as RCDC categories, research categorization, research condition and disease categorization, RCDC. And there's a website with these 200 plus areas of research into which our projects are categorized, and you can select one of these categories from this list under NIH spending category. Now, these categories are created at the end of each fiscal year, so it's important when you're selecting one of these categories to make sure that you've selected one of the past fiscal years. So the most recent fiscal year that ended was 2011, so we have categorization for 2011 and earlier years. If you try to search 2012, you won't be able to search on these different categories. In addition to the query screen, we have right next to it a new tab that we call Browse. And that's if you don't want to create a query and you just want to browse through the portfolio of NIH projects, you can go here and you can, this may be blocked by the chairs, but you can select NINDS as the institute you want to browse, and then you click Submit. And then you can browse through all of the investigators that NINDS supports, all the organizations. So there are about 3,600 different investigators currently being supported, 500 from 513 different organizations. These are the funding mechanisms that are used, the activity codes, which is the type of grant award, and you can look by location, the state and congressional district where the research is being conducted. And then you can click on any of these numbers over here to drill down and find out what those projects are in any of these categories. Whether you do a query or you do a browse, what you'll end up with is a project hit list. And this is what the hit list looks like, and there are several tabs across the top here. The first tab is simply the projects, a list of the projects that match your query. I mentioned publications. Each of our projects are linked to publications that have acknowledged support from that research. I'll give you some examples of that. Also, similarly with patents, all of our investigators, if they've submitted a patent application, are asked to report that to the NIH, and those are linked to the grants that have supported those patents. We have links created between our research and clinical trials in clinicaltrials.gov, and those are available on the Clinical Studies tab. A Data and Visualize tab lets you get a summary of your search results, and I'll give you an example of that in a minute. So if you did a search, text search on some disease, and you want to look at what funding has been like across fiscal years, or which organizations are receiving the most funding, you can look, get summaries of that on the Data and Visualize tab. We do maps, a geographical mapping. We have a Links tab, which is a link out to other tools that other people have developed that we haven't developed. We make this database publicly available in the hopes that people will develop their own tools for searching it. And we have, people are starting to do that now. We have one tool I'll give you an example of that's on the Links tab. And then the most recent addition is the News and More tab. In the past, all of our data and all the information we've provided has been very oriented around the grant and been very administrative in nature. The News and More tab is a way that we're experimenting with trying to link our research to other sources of information on the research, many of which is written in a more lay language, so it's more understandable to members of the public, and I'll give you some examples of that as well. So here's, I hope you can see this. This is an example of one of the research projects in the hit list. Each of the project titles here is linked to an abstract describing the research. And each investigator's name, if they've entered a profile, and we allow our investigators to come to the website and add their own profile links. So they can add a link to their institution homepage or their lab website, or if they have an institutional profile, they can link out to that. So this investigator, Dr. Eisenman, has added his website, his lab website, to Reporter, and you can link out from Reporter to his website and get more information on the research he's conducting. And then finally, there's this tab over on the far right, which is a Find Similar Projects tab. So if you click that little icon, it'll bring up 100 other NINDS projects that match that project by Dr. Eisenman. So these are 100 projects that are similar based on the text in their abstracts and other parts of their application. The Publications tab is a list of all the publications that are linked to one or more projects in the project hit list. So the projects I was showing you was active NINDS research grants. And these are 2011 publications, over 6,000 publications linked to those research projects. And each of these, if the title is hyperlinked, that means that there's a full text article available there, and you can click on the title and retrieve that manuscript. All of the journal references, the citations, are linked to PubMed abstracts. And again, just like with the Projects tab, if you want to find similar publications to this one, you can find similar publications in PubMed or Google Scholar, and you can even find other articles that have cited this article to find similar research. These are clinical studies are clinical trials that are linked to NINDS projects. So you have the project number, a clinical trials ID, and then clicking on the title of the study will give you a link out to clinicaltrials.gov to find more information. And we also show you what the status of the trial is, whether they're recruiting, active, not recruiting, and so forth. The Data and Visualize tab gives you a summary, as I said. So you can do summary by which institute is administering the research, what organization is conducting the research. So these, again, are active NINDS projects. These are the top, you'll get a list of the top 100 organizations funded to conduct that research. NINDS comes out at the top because Reporter contains not only extramural projects but intramural. And then you can use it on this table that accompanies the chart. You can drill down to see what those projects are and get a list of all of those projects. You can also do summaries by funding mechanism, distribution of projects by whether they're training grants or centers or individual research projects, and again, drill down and see what those projects are. On the Visualize tab, you also get we're also experimenting with new ways of searching the database using visual tools. So this one's kind of fun. It's called a product called Circles, which takes all of the text in the project abstracts and creates a hierarchical organization of all the terms that appear in there. And don't ask me how it does it. I don't really understand the details of how this works, but it's taken all of NINDS's active projects and created a hierarchy of terms that lets you drill down. So I can, for example, highlight brain injury here. Each of these dots is a different project, and it's highlighting the different projects that refer to brain injury. And then I can double click on brain injury and expand it. And under brain injury, there are these various different subcategories. So neuronal death is a term under brain injury, and again, these projects are highlighted, and I can click on one of these dots and bring up the project that's referring to. So it's kind of a fun way of exploring just another way of exploring the portfolio. It's kind of addictive. You can kill a lot of time playing around with that. And we're exploring new tools for doing similar kinds of things. It's just another way of searching. Geographical maps, we can show a heat map of NINDS's funding across the U.S. I hope there's no one here from Idaho. And then you can drill down for any specific state. You can see different dots for the different organizations we support. You can click on those and see, for example, at Tufts Medical Center, these are the active projects, and then you can drill down on any of those projects and so forth. And the highlighting here is congressional districts. So it'll show you heat map of funding by congressional district. The links tab I mentioned are tools that other people have created using these data, and I mentioned one in particular because NINDS helps support this project. It's NIHmaps.org. What they've done is they've taken the NIH research portfolio and did text mining to identify the different topics that underlie all of those projects and create what's called a topic map. And so these are all of these different colored dots are all of NIH's research portfolio mapped on this topic map, and you can then see where NINDS's projects fall on that map, and you can drill down and find out more about, click on each of those balloons, find out more about the project, find similar projects, and so forth. And then finally, news and more, which I think is really one of the more innovative things that we've done. And again, this is something supported in part by NINDS and several other IC institutes and centers at NIH have provided funding for us to do this, where we're now taking press releases and other documents that have been written about NIH research and linking them to the underlying projects that are being written about. So in the past, NIH has written a lot of reports on the research we support. It's never been clear, sometimes in books an inch and a half thick, that we're never sure, probably reside on someone's shelf somewhere, and we're not sure if anyone ever reads them. But now we can link those text documents back to the research that they are discussing and make it much more useful. So if you click on press releases, you'll get a list of 36 press releases that are referring to NINDS supported research. And if there's journal articles mentioned, you can link out to those. You'll see the grant number, the title of the news release, and you can click on these. And some of these are from Eureka Alert. We have many of our institutions subscribe to this news service, and they now can enter, when they enter a press release into Eureka Alert, they can also add the grant number along with it. So institutions are starting to do this so that we can take the news feed from Eureka Alert and link it back to the projects that are being discussed. Similarly, we have NIH written documents. NIH Research Matters now are linking to the projects as well, and even press releases from some of the institutions themselves. Websites are, you'll find links here to those. Eventually what we hope to do is make this kind of information searchable as well. The advantage of some of this is it's written at more of a lay language, so it's in terms the public can understand, not our technical abstracts, and then you might be able to search this as well. Finally, My Reporter. If you sign up for a My Reporter account, what that'll let you do is to save queries and, in particular, receive email alerts when new information is added to the database that matches your area of interest. So, for example, I've created a query for all of the awards that were awarded under this solicitation for novel therapeutic targets for Huntington's disease. And I can add a project and a publication alert, which means that any new project that's added to the database or any new publication that acknowledges support from one of those projects, I'll get an email, a weekly email that's showing these are the publications that match your alert, and then you can click on any of these publications in the email and pull up the abstract and see what it's all about. So any query, any favorite query you have, you can save it, sign up for alerts, and find out when new things have been added to the database that match your interests. And that's all I have. And Thank you.
Questions for Jim?
Yes.
Are there historical data in there that prior to the birth of report that are in this database, especially links between projects and investigators and publications?
Yes, the projects go back to 1972. Oh, I'm sorry. You can download data sets back to 1972. In the reporter query tool itself, we go back to 1985, and publications back to 1985. So it's about, what is that? Almost 20 years. We don't, the latter question being, can people use these links? These links are out there publicly available already. We're not creating any new links. We're just linking out to information that's already out there, so anyone can reuse it. As far as sending us new links, we don't have a way to do that now. What we're been suggesting is to get in contact with the communications office of the relevant institute or center, and they can enter those.
And the key point here is, if it is supported by the NIH, put the grant number in your release. Then they will automatically pick it up. We don't have to worry about being in touch.
Mm-hmm.
Other questions? Great. Thanks so
Okay,
much,
thank you.
Jim. I would also like to say that the NINDS website for quite some time has had a section which lists a lot of press releases from research institutions and universities and medical schools of research that was funded by NIH. It makes for some pretty interesting reading, and doesn't always mention the grant number, but it's done in lay language, and half an hour spent perusing what we funded, and the lay language summaries, I think, can give you a pretty good idea of the breadth of the things that we do. And this is in addition to the NINDS press releases and news stories. So it's what the universities thought their investigators were doing that was worth a picture and a write-up. So we have a time for a short break. And then when we come back, and probably why don't we reconvene at 10 past, breakout number 1 will be in this room, and breakout number 2, which is SBIR, STTR, and Programs and Opportunities to Advance Therapeutics, will be in A1, A2, which is the other side of the lobby. And I will not be offended, nor will Alan, if the majority of you go, even if all of you go, to the SBIR, STTR, Programs and Opportunities to Advance Therapeutics. The reason for having the NIH 101 is if there are new voluntary groups or people who are here for the first time, we thought it might be good just to have a pretty open question and answer. We have some slides, but it's mostly for you to ask questions about how to work with NIH and how does NIH actually itself work. So 10 past. Thanks a lot.